Good afternoon, everybody, and welcome to the latest GARDP Revive webinar on Efflux inhibitors, a strategy to tackle multidrug resistance. I am Laura Piddock, Scientific Director of GARDP, and I will be moderating and hosting this webinar with three guest speakers, Helen Zgurskaya, Ruben Hartkoorn, and Tim Opperman. So Revive is our education and outreach activity, and Revive webinars capture essential research and development technical knowledge, and we share with everybody expertise such as that of the three speakers today. The aims of the Revive programme are to facilitate learning, connect people together, and share old and new knowledge and wisdom. So we do this through a variety of activities. As you know, today we have Revive webinars. We also have AMR discussions. All of our webinars are recorded and they are on YouTube and freely available to everyone forever. It's important to note that there are also subtitles. So if you wish to use subtitles today, please switch on via the YouTube offering. We also publish antimicrobial viewpoints, and just wish to draw your attention to our latest viewpoint, which is by one of our speakers today, and the link to this will be in the chat in a minute. We publish between 10 and 12 viewpoints over the year from a variety of different speakers on a variety of different topics. I do hope that you'll enjoy them, and please see the link to this part of the Revive website. We also have a variety of resources, and one of the ones that is proving most popular is the Antimicrobial Encyclopedia. And this is just a screenshot of part of the encyclopedia. And if you click on any item within it or search, you'll find much more information, including links to other information and other websites, such as the FDA, for example, and also some videos recorded by Revive experts for us that further explain the definitions. So do take a look at this resource. So on to today's webinar. As you all know, we have a live Q&A after each of the three speakers has spoken. So this will be at the end. But if you wish to start asking questions during any presentation, please submit as shown here on this slide. Please indicate the name of the speaker to which you are addressing your question, so that it's easy for me to identify the person when I ask it. So please do submit your questions throughout the webinar, and I hope that you find this stimulating. So these are our speakers today. We have Helen Zgurskaya. As I mentioned, she will speak first, followed by Ruben Hartkoorn, and then Timothy Opperman. So first, I wish to welcome Helen. She is a George Lynn Cross Research Professor in the Department of Chemistry and Biochemistry at the University of Oklahoma in the United States. So firstly, thank you, Helen, for getting up so early to do this webinar. She's a fellow of the American Academy for Microbiology and leads a research program on antibiotic resistance, multidrug efflux mechanisms, and efflux pump inhibitors. She's written numerous papers and helped to prepare and co-organise several conferences. She's an associate editor of ACS Infectious Diseases and a member of the editorial boards for journals Antimicrobial Agents and Chemotherapy and Frontiers in Microbiology. So welcome, Helen. I look forward to listening to your presentation.
Thank you so much, Laura, for a very kind introduction. Hi, everyone. It's my pleasure to be here today and tell you about our recent studies on efflux pumps in bacteria. So all of you are familiar with the bacterial cell envelopes, which are the primordial resistance mechanism of bacterial species. So although structurally these bacterial envelopes are highly diverse, they follow the same physical, chemical, and biological rules. So these rules are to allow nutrients to get into the cells so cells can grow, and then all bacterial cells will have some kind of permeability barrier for toxins, and these passive permeability barriers will be reinforced by active efflux pumps. So we tried to figure out how this protection barrier works, and we came up with several rules that I want to share with you. The first rule is that efflux, active efflux synergistic with the limitation only when across the same membrane. And this applies to both the outer membrane and cytoplasmic membrane, depending on the species and composition of the efflux pumps. You can see the synergistic interaction between permeability barrier and efflux, for example, in gram-negative bacteria. If you permeabilize the outer membrane, or if you inactivate the efflux pump, or if you combine the two mechanisms, the permeation across the outer membrane and inactivation of efflux pump in one species, in one strain. So what will happen in this case? So you will see, if you compare the wild type cells with double compromised cells that lack the outer membrane barrier and efflux, you might see several orders of magnitude change in MICs of antibiotics. However, these changes in MICs of antibiotics will be also species specific. In some bacteria, such as E. coli and Acinetobacter, you will see significant changes in novobiotine. Then if you go to other species, you can also see changes in susceptibility to azithromycin or gentamicin. So you can also see the changes in permeability barriers and the synergistic interactions between efflux and the outer membrane if you look at the kinetic of intracellular accumulation of fluorescent probes. So here you're looking at example of E. coli, and you're looking at these 4 types of bacterial strains. The wild type, you see very little accumulation of the probe inside the cell. If you inactivate efflux, so you will see some increase, but the rate will be kind of slow. If you permeabilize the outer membrane, you suddenly see significant increase in accumulation. And of course, if you double compromise the cell, you will see even further increase in intracellular accumulation. So the rule number 2 is that cells often produce different type of efflux pumps, and they can be either synergistic or additive with the transporter acting across the same membrane. So if you look at this figure here, right? So this transporter, which spans the entire envelope of gram-negative bacteria, can pump drugs across the outer membrane. So if you have these 2 efflux pumps co-express, then you can expect to have additive effects on susceptibility to antibiotics. However, if you co-express the pumps that are acting across 2 different permeability barriers, such as MDFA of E. coli and ACRA-B12C of E. coli, in this case, you can expect to see synergistic interactions between the 2 pumps and multiplicative changes in susceptibility to antibiotics. So one of the paradox, however, is that if you look at certain species, such as Acinetobacter baumannii, for example, you will find that in multidrug resistant strains, actually, the same type of pumps are often overexpressed, such as AD-ABC, IJK, or even AD-FGH. All these 3 pumps act across the outer membrane, and nevertheless, they are selected by antibiotic exposure to be co-expressed in MDR strains. So the explanation for this comes from the rule number 3. So Acinetobacter, as well as many other MDR strains, also express additional mechanisms of resistance. And one of those mechanisms are enzymes that modify or degrade antibiotics that get inside the cell. So these enzymes actually, in kinetic biochemical terms, they're similar to efflux pump because they reduce effective concentration of antibiotic in different cellular compartments, either in the periplasm, such as beta-lactamases, or inside the cytoplasm, such as, for example, transferases. So what happens in this case, and we showed this for Acinetobacter, is that enzymatic inactivation of antibiotic can also be either additive or synergistic with efflux and transmembrane diffusion. And of course, additive contributions will be if enzyme and efflux pump reduce the concentration of antibiotic within the same compartment, such as beta-lactamases and pumps that act across the outer membrane. Whereas if, for example, efflux pump and enzyme reduce the concentration in 2 different compartments, then you can expect to have synergistic interactions and multiplicative changes in susceptibility to antibiotics. So my first take-home message is that when you think about efflux pump, think about cellular, intracellular context, because efflux pumps in combination with diffusion barriers, enzymatic inactivation can generate either additive or multiplicative changes in intracellular concentration of antibiotics. And of course, this will affect their antibacterial activities. So we will focus now on one specific efflux pump. The reason for this is that R&D, or resistance modulation division, efflux transporters are the major contributors to clinical antibiotic resistance in gram-negative species. So these pumps are 3-component efflux machines. So the inner membrane complex contains the transporter and periplasmic membrane fusion protein. This inner membrane complex recruits the outer membrane channel that allows the pump to pump drugs across the outer membrane. So we have a lot of structural information about these pumps, and we also have very nice computational models that reflect on properties and also dynamics of these pumps. So one of the major characteristics of these R&D pumps is that they're very polyspecific. So the polyspecificity comes from several different mechanisms. The mechanism, of course, number one is that the pumps are actually synergistic with barriers and enzymes, those rules 1 through 3 that I described. So in many cases, when pumps act in the context of diffusion barriers and enzymatic inactivation, they seem so powerful and so polyspecific because of the synergistic interactions. In addition, there are certain structural features in these pumps which are very important for polyspecificity. They have 2 drug binding sites. They have multiple channels. All these binding sites have low affinity entry, and water-mediated translocation plays also a very important role in this polyspecificity. Of course, the subject of this webinar is, can we inhibit these pumps, and what are the best strategies? And you can inhibit these transporters at different steps of assembly and drug transport. Several classes of inhibitors have been identified. And of course, all these classes of inhibitors also affected by the same rules that I described above. And I'm going to talk a little bit how we actually can distinguish what molecules will be the good efflux inhibitors. So when we looked at a series of different antibacterial compounds, what we found is that all of them have a spectrum of recognition by efflux pumps. So in any series that you can take, you will find good substrates of efflux pumps. Some of them might be inhibitors of efflux and others will avoid. So this brings us to the paradox number 2. So there is no clear distinction between efflux substrate inhibitors because the same chemical scaffold can present these very different properties. So this also comes from structural studies and molecular dynamic simulations. So as you can see, this is a summary of structural analysis of ACRB and MEX-B from Pseudomonas aeruginosa. And the typical characteristic feature is that large antibiotics you will primarily find in the structures bound to their excess pockets shown here in the blue. Some of the drugs you can find both in access pocket and also in distant pocket, which is shown here in green. And inhibitors typically are more localized to the distal pocket than to access pocket. So the questions that we asked is, what is the difference between efflux avoiders, substrates, and inhibitors? So for these purposes, we set up an experimental pipeline which starts with the assembly of chemical library of antibacterials and efflux pump inhibitors. So our current library is about 1,300 compounds. And the library is analyzed in the set of experiments. So the first is analysis of susceptibility to antibacterial activities of these compounds in 4 strains that I described. So we're looking at wild type, hyperparinated cells, efflux deficient, and double compromised. And then we're looking for efflux inhibitory activity by doing several different assays. One of the assays is to look for accumulation of fluorescent probes that I already showed. And another is to look at potentiation of antibiotic activities. So from these experimental studies, we derived several efflux ratio, several values, several numbers. Total barrier ratio, it is a ratio of activities in wild type and double compromised. Efflux impact ratio, we're looking at hyperparinated cells with and without efflux. Outer membrane impact ratio, we're looking at efflux deficient cells with native membrane and hyperparinated. And inhibition ratios can come from either fluorescence or from potentiation. So the second question is, what properties of compounds do correlate with antibacterial and EPI activities? So we're looking for 3 sets of numerical properties. So first is quantitative structure-activity relationship. In this case, we're looking at chemical structures of compounds and their quantum mechanical properties. So these properties of compounds themselves then interact with features in the cells, right? With the outer membrane, for this, we're looking at interactions of molecules within different layers of the membrane. And they also interact with efflux pumps. So we are also looking for the interactions between all these compounds with access pocket and distal pocket in the transporter. So the rest of the slides will be focused on this particular transporter, MEX-B-PRM, and outer membrane of Pseudomonas aeruginosa, so which is one of the toughest drugs, toughest bacterial species in clinics. All this data, experimental and also computational, then go into machine learning. So the first data set is about permeation across the outer membrane. So for the drug to get to the efflux pump, they must be able to cross the outer membrane. So we looked at Pseudomonas aeruginosa again, and the model that we generated gets very nice prediction accuracy, about 96%. And the strong permeators we found have certain features, and those features are hydrogen bonding with different layers of the outer membrane. They have hydrophobic, certain hydrophobic surface area. Flexible molecular scaffolds are important, and one of the interesting, important features for these permeators are interactions with deep pocket residues in MaxB. So good and strong and weak permeators have different numeric values for these descriptors, which helps with design of compounds for permeation across the outer membrane. So the question was, of course, why interaction with the pump contribute to permeation. So we looked at more focused series compounds to develop to understand the features of efflux avoidance and recognition. And we used Rempex compounds developed by Microcyte and then Rempex and currently QPEX. Olga Lamalskaya from QPEX donated the compounds for us to study. And we developed models that, excuse me, that distinguish avoiders from inhibitors. And efflux avoiders we found have low or no contact with access pocket of bacterial pumps. They have hydrogen bond acceptors and also hydrophilic low log D. So, and again, as you can see, the interactions with the pump play a very important role in distinguishing between efflux avoidance and inhibition in this case. So one particular feature of this compound is that they permeate the outer membrane of Pseudomonas very well because of this particular positively charged group. And also, again, the series contains the entire spectrum of interaction with the efflux pump. So to look closely why this interaction with pump is so important, right, for generating different models, we took a closer look at how these Rempex compounds interact with MaxB of Pseudomonas. So here are 4 representative compounds. One of them is a substrate and does not inhibit efflux pump. 2 compounds are efflux inhibitors, but they differ because one of them is recognized by the pump as a substrate, whereas another one is not recognized by the pump as a substrate, but both potentiate the activities of antibiotics when MaxB is expressed. And then this 4th compound is actually a avoider of efflux pump. So we looked at amino acid residues in axis and distal pocket. We mutated those residues, and again looked at the details of interaction of these different classes of compounds with MaxB pump. So machine learning then was used again to figure out what exactly is going on with the entire series, and the results are shown here. What we found essentially is that these different classes of ligands within the series, they efflux avoiders from ligands are distinguished at the level of access pocket. And although some of them progress to the interface shown in blue here, in interface, we already see the divergence between efflux inhibitors and efflux substrates. So the interface is a point of branching for these 2 classes of ligands. And then in the distal pocket, we can see more specific interactions between ligands that engage polar and charge residues, and these ligands are typically substrate and are effectively expelled, whereas substrates that engage hydrophobic and aromatic residues typically slow down and hinder the transporter and can act as inhibitors. So my conclusions are here, and my messages. We have now integrated antibiotic permeation and resistance model that nicely explains laboratory and clinical observations. We have different experimental tools to look at contributions of efflux, permeation, and other mechanisms of resistance. And we can clearly separate these contributions in antibacterial activities and biochemical activities. So efflux pumps can make sense only in physiological environments, and this is something to keep in mind. Efflux ligands have different physical, chemical, and molecular level properties, and those properties are distinct from avoiders. Ligands fit into the spectrum depending on their physical, chemical structures and properties, and permeation, efflux avoidance, and inhibition models are now getting better and better and are predictive of such properties among unrelated compounds. Oops, sorry.
Thank you, Helen.
My slides are stuck. Sorry, just one more thing. Sorry, it's important. I would like to thank all members of my lab, present and past, all my collaborators and collaborators from industry and NIH for funding, and thank you so much for your attention.
Thank you, Helen. Excellent overview and of your recent data and indicating some of your publications as well. So that's been excellent. So we will now move on to our second presentation from Ruben Hartkoorn. He leads a research team on antibiotic drug discovery and development and innovation at the Institut Pasteur of Lille in France. His group focuses on discovering and developing novel approaches to combat bacterial infections, particularly focusing on novel classes and improving the penetration of antibiotics into bacteria. He received his PhD from the University of Liverpool in the UK after working on eukaryotic efflux and influx pumps, and also on HIV and tuberculosis. And before he joined INSERM in 2015 and launching his group, he carried out postdoctoral research on rifampicin tolerance at EPFL and investigated drugs active against toxoplasma and malaria at the University of Geneva in Switzerland. So over to you, Ruben. Thank you.
Thank you very much for this kind introduction and for being invited to present our work on the discovery and development of new efflux pump inhibitors and understanding their mechanism of action, and with really the aim of identifying tools to fight the constantly increasing MDR species that are becoming clinically relevant. As mentioned, I have a research team at the Institut Pasteur of Lille in the north of France, and there's a little bit of a lag. Our team focuses on fighting AMR through different strategies. Firstly, by trying to discover new molecules that have new targets and therefore can target drug-resistant bacteria. We have an ERC-funded program on trying to improve antibiotic penetration into bacteria by coupling antibiotics through a biomimetic manner onto siderophores and therefore generating Trojan horse-based antibiotics. And the topic of today is our attempt to improve our ability to kill gram-negative bacteria by preventing antibiotic efflux through these broad-spectrum R&D type efflux pumps. As was very nicely already introduced in Helen's presentation, the membrane of gram-negative bacteria is incredibly difficult to penetrate because of penetration issues and because of efflux. And this is really the main reason why it's so difficult to develop new antibiotics against these bacteria. So the bacteria is able to efflux molecules through narrow spectrum pumps, but it's really these broad spectrum pumps that causing most of the general problems we have in drug development, and of course in the clinic when it comes to the acquisition of increased drug resistance. These RMD pumps are expressed in many gram-negative bacteria, if not all. And most of them have many different versions of them. However, there are some predominant pumps that are typically responsible for the efflux of most of the antibiotics. And in E. coli and Klebsiella, which is the main focus of this talk, these are the ACR A, B, Tol, C systems. And these systems are really responsible for preventing most antibiotics entering, especially when it comes to drug discovering new antibiotics. And on top of that, mutations in transcriptional regulators are clinically present that cause clinical resistance because they cause upregulation of these pumps and even increased efflux. So as was mentioned, these pumps are made, these R&D type pumps are made of three components: an inner membrane pump, and then an adapter, ACRA in this case, and an outer membrane channel. And these parts come together to link the inner and outer membrane. And the parts of each part is made of three components, an asymmetric trimer. So that is to say that it's made of three copies that look different. Each protomer has a different configuration, and that in the case of E. coli and Klebsiella is very important for its mechanism of action. So the loose protomer here in blue has an open access pocket or proximal pocket where antibiotics or other xenobiotics can enter either from the inner membrane or for the periplasm. And when they enter, they can then have access. There is a conformational change that is thought to happen where the protomer is changed to a tight configuration, the T configuration, where a deep binding pocket is also accessible. When that is accessed, the proton motive force on the inner membrane allows for the final configuration change that converts the tight protomer to the open protomer where the proximal pocket is closed, so there's no more access, but then other pocket is open that allows the diffusion and exit of the antibiotic through ACRA and Tol C in the outer membrane. So clearly, the broad spectrum of antibiotics that are pumped by these pumps and the efficiency at which they have managed to do this make this protein, this system, a nuisance for drug development. And thanks to the efforts of our colleagues here and many others, many different inhibitors have been described thus far. And this is a non-exhaustive list, and sorry if I have not included your favorite inhibitors, but most people will know PA-beta-N, a very useful and accessible inhibitor that we can use to inhibit many R&D type efflux pumps. But we also have the pyridopyrimidines and the pyranopyridines, which will be covered in the next talk, which are incredibly potent and really effective inhibitors that bind to this deep binding pocket that was discussed in the previous talk. And these are really fantastic molecules, but they bind in this pocket that is very hydrophobic, and that has sometimes the problem that you come up with molecules that are themselves hydrophobic and have characteristics that are sometimes difficult to convert to drug-like properties. This is often possible, but it takes sometimes a bit more work. And we asked ourselves whether it would be possible to come up with alternative scaffolds of inhibitors that were more water soluble and had more drug-like properties and perhaps work through another mechanism of action. So that was really our starting point. And when we want to do some discovery of new scaffolds, really the main important question is, what are we going to test? Because we're not going to discover anything other than what we're going to test. So this all started with a fantastic collaboration with the Faculty of Pharmacy here that's also associated to the Institut Pasteur of Lille, particularly in the beginning with Nicolas Villon, where we thought that the interesting starting point would be to screen small fragment molecules that have very high water solubility, but because of their size probably have low affinity for any particular binding pocket. So we decided to test them at a very high concentration of 300 micromolar. And we tested them just by themselves, hoping that they would not have activity by themselves, but also in the presence of a ACR-AB Tol-C substrate. In our case, we tested that with the natural product antibiotic pyridomycin, but this could easily have been done with another substrate. And the idea was to find those inhibitors that were active only in the presence of a sub-MIC concentration of pyridomycin. And we were a bit lucky because from this very small library of molecules, we did identify a compound, a pyridopiperazine, which had the features that we were looking for. So it had no activity by itself, but as you can see here on the right, in the presence of pyridomycin, it had a dose-dependent activity, and therefore some level of synergy was being hinted at. So that, of course, does not mean we target the pump, but we had some validation that we did. And the first step we did is we had a look at whether we could boost pyridomycin activity when we genetically knocked out the pump components, and this was no longer the case. So that was supportive. And also we showed that this boosting was not specific to pyridomycin. We were able to boost a lot of well-known efflux pump substrates, such as erythromycin, tetracycline, and ciprofloxacin, but other antibiotics like like aminoglycosides that are not substrates were not boosted. So this was supportive, but not really indicative of the exact mechanism. So really a crucial experiment we then managed to do, and this was not easy, it took a lot of effort, was to try and identify by spontaneous manner mutants that were resistant to the boosting effect of this pyridopiperazine. And we actually managed to do that by having a selection on a combination of erythromycin and the pyridopiperazine and adding 10 to the 9 bacteria to this system. And this allowed us to identify a few mutants, which nicely had no altered sensitivity to the antibiotic itself. They also could still be boosted by PA-beta-N. However, the addition of the pyridopiperazine itself had no impact anymore. So this is really the phenotype we were looking for. And thanks to whole genome sequencing and validation by reverse genetics, we could confirm that the causative mutations that were causing this resistance were in a new location in the transmembrane domain of ACRB, a region that had previously not been described to be associated with any type of inhibitor. So as you can see here, it's in helix 5, I believe, of the ACRB protein. So in parallel in this program, we have fantastic collaboration still active with Marion Flippo, who is driving all the medicinal chemistry and SCR studies in this work. So we started from this initial hit, which was very tractable, and we defined very rapidly that the nitrogen on the piperazine had to be basic and had to be more or less maintained as it was, and that the chlorine on the molecule was very beneficial for activity, could be modified for other halogens, but otherwise needed to be there. But at the bottom of the molecule, we could make a lot of modifications, and this allowed us to really improve the compound quite rapidly, initially to this quinoline version you see on the right, that had a 20-fold better activity. And by having a better molecule, it also makes everything a bit easier. We quickly showed that this molecule was able to boost a whole panel of antibiotics that are known to be substrates for efflux, and we were able to boost them to the same level as when we knock out the efflux pump, which was really reassuring, and we were not able to boost them any further, which is also important. So really showing that we were staying on target and not off target. We could do checkerboard assays, and also very importantly, the data we saw on E. coli, we could reproduce more or less on Klebsiella pneumonia, where a very similar ACR-AB Tol-C system is present. So we are equally active on both systems here. Thanks to this more potent molecule, we could also work with our collaborators at the Goethe University in Germany, in Frankfurt, to get a co-crystal structure. And this allowed us to identify the binding pocket. And very happily, we saw that this binding pocket was very close to the mutations we had identified in the transmembrane domain. And very interestingly, we only found density for the inhibitor in the L protomer, the loose protomer of the trimeric structure, showing that there was some kind of specificity, particularly for this protomer. When we look carefully at the binding interactions, we were very happily surprised to identify that this critical basic nitrogen of the piperazine was interacting with aspartic acid 408. This is a critical aspartic acid involved in the proton relay of the ACR-AB-Tol-C pump. And we believe that by interacting with this residue, we are probably altering its ability to use the proton motive force to generate the energy for efflux. So really an allosteric mechanism of action. We also noticed that at the bottom of the pocket there was some more space, and the medicinal chemists in the group of Marion were very rapid to exploit this space and try to find additional interactions to improve the activity of these molecules. She did this both, she and her team did this both on the pyridine series and the quinoline series, identifying molecules that were much more potent, but some of them not having all the perfect drug-like properties. And in the case of the molecule here on the right, it's a quinoline analog that looks very similar to the original, has a similar activity, potency, but it had much better drug-like properties, which I'll get to in a bit. Thanks to our consortium that we had built at that time, we also had the chance to work with Achilles Frangakis and his very talented PhD student, Clara Bornsen, who were able to do the cryo-EM co-crystal structure now of this improved quinoline pyridylpiperazine. And we could show that the interactions that had been designed by Marion and her collaborator and her team were indeed happening. We could form these additional interactions with acidic residues at the bottom of the binding pocket. Again, as we saw in E. coli, here in this structure of ACRB from Klebsiella pneumonia, we saw that the inhibitor was only present in the L protomer, and that was all consistent. So at this point, we started to think about, what can we do with such molecules when it comes to AMR? And our question was really, first of all, can we inhibit any resistance that is caused by overexpression of ACR-AB-Tol-C? And this in Klebsiella typically occurs when there are mutations in RAMR, a transcriptional regulator that causes overexpression of this pump system. And in these two checkerboards with the wild type on the left and the RAMR mutant on the right, this is Klebsiella, you can see that these RAMR mutations cause tenfold resistance, more or less, of levofloxacin, where it goes from 63 nanogram per ml MIC in the wild type to around about 500 nanogram per ml in the RAMR mutant. Nonetheless, when we now, on top of that, add these efflux pump inhibitors, in both cases, we can fully sensitize these bacteria to levofloxacin down to a level of 16 nanogram per ml, meaning that we can overcome this additional resistance mechanism. Although it has to be said that we need more compound in order to achieve that level of inhibition, which is self-evident in a way, because there is more pump, there's more target to be inhibited, therefore we need more compound. And once we get more potent molecules, this will be less of a problem. So this molecule was chosen, as I mentioned, not necessarily because it was the most potent, but it had features that made it possible to use it for in vivo proof of concept. So, and particularly, its microsome stability was reasonable, was very good, in fact, meaning that it would not be eliminated directly once we administer it to mice. These molecules were found in mouse PK studies to be orally bioavailable. And indeed, that the plasma levels were reasonably stable. There was a low level of clearance of these molecules, as you can see in the PK profile on the right. We did notice that the level of the drug found in the plasma was actually not that high. But when we looked at other organs, and particularly the lung, we found there was a massive accumulation in the lungs of these pyridyl piperazines. We also looked at the lining fluid of the lung, the ELF, and we found a more or less intermediate concentration there. And this gave us the belief that we should, of course, use a lung infection model for our initial proof of concept. So that's what we did. We worked with Laurie Vermel here at the Center of Infection Immunity of Lille at the Institut Pasteur. And we decided that we wanted to look at lung infection of Klebsiella pneumonia and hypervirulent strain. And we decided to look at whether we could boost the activity of levofloxacin. So in this graph, you can see that in blue, in black first, we have the untreated group, and next to it in blue, we have the group of mice that were treated only with the efflux pump inhibitor at 30 mg per kg orally, and we see no difference. So the compound by itself had no impact on the infection. Of course, when we added levofloxacin at 10 milligram per ml, and this is IP, we had a great reduction in the bacterial load in the lungs. But when we, on top of that, we added the efflux pump inhibitor, this bacteria, and there was a significant, around about 1 log additional decrease in the bacterial load, really confirming that there was a significant impact of this efflux pump inhibition. And it needs to be noted that this is on levofloxacin on a wild type infection, wild type strain infection, where levofloxacin MIC only becomes 4-fold better. So this was really showing us that we had a good efficacy, and now we need to explore further whether we can translate this to the reversion of resistance in MDR strains and look at other antibiotics, and also look at more potent pyridyl piperazine-based efflux pump inhibitors. So to conclude, I hope I've convinced you that we have a new chemical scaffold that acts very differently compared to the deep binding pocket inhibitors. I'm not saying better, but very differently and complementary. It has a novel mechanism of action, exploiting a novel pocket that is very interesting, because in TB there are R&D type efflux pump inhibitors that have inhibitors acting in a similar area, and they have been developed into the clinic, or being developed into the clinic. So this is a good pocket to target. We have a potent, we have not shown the most potent here, but we are getting very potent molecules now on Enterobacterialis R&D efflux pumps, although we have to say that currently the potency on other species such as Acinetobacter baumannii and Pseudomonas is a little less high. So we're a little less potent against them. We have now a favorable and tractable pharmacokinetics. We can modify that depending on the analogs we have, so we can match now our antibiotic PK with our efflux pump inhibitor PK. So that will be important for future progression of the molecule. Further optimization is going on, and we really want to try and demonstrate that we can use these molecules to impact clinically MDR strains. And this is really our aim in the future. So this work was done with a fantastic collaboration. I have mentioned the people here involved in the early parts. So this is people from my team at the CDUZL at the Institut Pasteur of Lille. Special thanks to Colin Play, Juan Carlos Jimenez Castellano, and Laurie, of course, as well as Elisabeth Pradel. This work was pivotal to have fantastic medicinal chemists on this, and this was provided thanks to the wonderful work of Marion Flippo and Nicolas Villon at the Faculty of Pharmacy. And we had fantastic collaborative fun with Martin Poss and Achilles Fangakis from Frankfurt, who did all the structural biology with us, as well as Attilio for molecular dynamics. And I will leave it at that. Thank you.
Thank you, Ruben, for an excellent presentation. We will now move on to our last presentation, given by Tim Opperman. He's Director of Microbiology at Microbiotix in Worcester, Massachusetts, in the United States. So thank you, Tim, also for getting up early to do this webinar for us. He's worked for over 20 years in this field and is the principal investigator on a project to optimize a novel series of R&D type efflux inhibitors. Before he joined Microbiotix, he worked at Genome Therapeutics Corp, and he used bacterial genomics to identify novel antibacterial targets, developed screening assays, and carried out high throughput screens for these novel agents. He received his PhD from Indiana University after working in the lab of John Richardson, and then did postdoctoral research at the Massachusetts Institute of Technology in the lab of Graham Walker. So over to you, Tim. Thank you.
Thank you, Laura, and thanks for the invitation to speak today. So I'm going to be talking about the hydrophobic trap, and this was touched on in the previous talk. I'm going to speak about it both as a target and also as a challenge for developing drug-like inhibitors of efflux pumps. So let's see if we can move this forward. All right, so I'll just go quickly through this slide. It's been covered in the previous two talks. So the gram-negative bacteria are intrinsically resistant to antibiotics because of limited influx of antibiotics due to the outer membrane and selective porins, and by impressive arsenal of efflux pumps that have several classes that pump drugs from the cytoplasm to the periplasm, and then this class of pumps, the R&D pumps, that finish the job and move these drugs from the periplasm to the outside of the cell. So these R&D pumps, as you heard, play a major role in intrinsic resistance, and overexpression of these pumps can lead to multi-drug resistance. This won't go. Okay, there we go. Having trouble here. Okay, so that makes the R&D pumps fantastic targets for dealing with multi-drug resistant bacteria. And it's known that if you inhibit the pump, end up with increased antibiotic susceptibility. Inhibition of the pump also decreases the frequency of resistance to mutation and acquired resistance, and also affects virulence, especially in the gut and where there's the bile acids and also biofilm formation. So the idea then would be to use a R&D pump inhibitor as adjunctive therapy with an antibiotic to improve the activity of the antibiotic and prevent resistance. Just a brief history of efflux pump inhibitor development. I'm just focused on early development of efflux pumps that was initiated by a company called Microbiotix back in the late 90s, and which changed names several times over the years. But they started by developing a series of peptidomimetics based on the PA-beta-N scaffold, but these were abandoned due to kidney toxicity, and also a class of pyridopyrimidine inhibitors that were developed in collaboration with Daiichi, which culminated in this compound D13-9001, which is, as far as I can tell, is no longer being developed. But I think both of these compounds are not being developed, not because of the target, but other issues such as toxicity. Okay. This is all right. Going the wrong way. Okay. Well, so in the early days, the question was always, how do you target these efflux pumps with inhibitors when they have a large polyspecific substrate binding site? And so this over here, there's a bunch of efflux ACRB substrates, and you can see they're quite diverse in nature, but they all share the similar characteristic of being hydrophobic in nature. And then the binding pocket, the deep binding pocket of ACRB is quite large. So it raised the question of how do you develop inhibitors, and the answer is the hydrophobic trap, which is the Achilles heel of R&D pumps, and or at least it's one of them. I think the previous talk described another one, so it looks like Achilles has at least two heels. Hopefully there are more. So the hydrophobic trap was discovered by the Yamaguchi group in Japan. They reported the co-crystal structure of this Daiichi compound D13-9001 bound to ACRB, and so this is a figure I stole from their paper showing D13-9001 in yellow binding to the site they call the hydrophobic trap, which is distinct from the binding site of two known inhibitors, minocycline and doxycycline, shown in blue and green. And they call it the hydrophobic trap because the site is lined with hydrophobic residues, either aromatic residues or aliphatic residues. And they also found that this compound is not a substrate of the pump. And further work by them showed that or strongly suggested that the compound works by inhibiting conformational changes that are required for pump activity, or the switch between the different conformations that was described in the previous talk. So we've been working on a class of efflux pump inhibitors called the peranopyridines. We identified these in a screen for compounds that potentiate the antibacterial activity of ciprofloxacin versus E. coli. And we identified our first hit, MBX 2319, exhibited potent activity against the Enterobacteriaceae, except Proteus, and very weak activity against gram-negative non-fermenters like Pseudomonas and Acinetobacter baumannii, mainly due to the inability of these compounds to penetrate the outer membrane. These compounds don't have any intrinsic antibacterial activity, with MICs are greater than 100 micromolar, the highest concentration we test. And they potentiate the antibacterial activity of almost all the compounds we've tested that are known pump substrates. So our initial chemical optimization of this series focused on improving the potency of these compounds. And we quickly found these two compounds, MBX31 and 3132 and 3135, that exhibit, they have a 30-fold improvement in potency in combination with either levofloxacin or piperacillin. And then so to determine how these compounds work, we were collaborating with Martin Pose's group and his talented postdoc at the time, Hanno Schutz, crystallized MBX 3132 and 3135 with ACRB. This is ACRB pair. It's a construct they made that comprised of just the paraplasmic domain of ACRB. And they showed that the MBX 3132 binds to the hydrophobic trap. And so this is another view of it, showing the residues that interact with the compound, along with their predictive binding energy. And so the darker the color, the more binding energy they provide to the interaction. And the two most important interactions were between phenylalanine 178 and phenylalanine 628. And another interesting thing is that the improved potency of these compounds is due to hydrogen bonding between this nitrogen group and a ordered lattice of water molecules in this open space here. Over here is just another view of the interactions between binding site residues and MBX 3132. And again, you can see the most important ones are between phenylalanines and these phenyl groups in the molecule. And we confirmed the binding site by making site-directed mutations of the residues involved in binding. So we altered each residue to either alanine, leucine, or another aromatic residue. And you can see that the residues shown in red inactivate the pump activity. The orange ones reduce affinity of the pump for the compound, and then the green ones are like the wild type. So these two phenylalanine residues are essential for pump activity. If we mutate this phenylalanine 628 to leucine, we can actually decrease the affinity of the compound for the pump. And likewise, if we mutate other surrounding phenylalanine residues like phenylalanine 615 and some of the other hydrophobic residues, we can also decrease the affinity for the pump. So using this, we were able to confirm the binding site and show that the hydrophobic interactions are essential for inhibition. Another piece of evidence for the mechanism of action came from this paper by Ben Luisi's group, where they reported a cryo-EM structure of MBX 3132 bound to the ACR ABZ TOL-C pump, which confirmed the binding site as well. But when they analyzed the pumps and the grids that they used to determine the structure, they found that 73% of the ACRB trimers were locked in this TTT conformation. So all the three protomers of ACRB were locked in the T conformation, whereas functioning pump you'd expect to be about an equal ratio between the 3 different conformations. So that providing evidence that MBX 3132 is acting like a monkey wrench thrown into a machine or a spanner thrown into the works. So then next, chemical optimization program, we focused on trying to synthesize a compound, an analog that we could use for a proof of principle experiment in animals. So you can see that the initial hit has some problems. It's not very soluble. It's not stable in murine liver microsomes, which means that the half-life in mice would be very short. With MBX 3132, we still have the same problem with stability, metabolic stability, but with the addition of this amido group, we also have the problem of inhibition of cytochrome 3A4. But we managed to synthesize this compound MBX 4191, where the amido group has been changed for a tetrazole, which improves solubility somewhat, improved the liver microsome stability, but still had CYP inhibition activity. But this was good enough for us to move forward and with a proof of principle experiment in mice. So this shows the results of a efficacy study we did with Bill Weiss group at University of North Texas Health Science Center. So this is a model of sepsis in CD1 mice, and the mice were infected with intraperitoneally with Klebsiella pneumoniae strain. It's case PC that's carbapenem resistant and also resistant to minocycline. We don't know the exact mechanism, but I suspect it's a tetracycline pump. And so the question was whether we can rescue the antibacterial activity of minocycline versus this strain in the murine sepsis model. So mice were infected and treated with either minocycline alone, the vehicle alone, or a combination of minocycline and MBX 4191, either at 100 mg per kg or 30 mg per kg, administered through the subcutaneous route. So mice treated with 50 mg per kg of minocycline are shown here, and you can see that the MBX 4191 rescued the activity of minocycline as monitored by survival. And you can see at a 200 mg per kg minocycline, both 100 and 30 mg per kg minocycline were rescued activity of minocycline. So these compounds actually work in mice, and so that prompted us to go on another round of optimization to try and make these compounds more drug-like. And so this was touched on in the previous talk, and it's kind of the second theme of my talk, is that compounds that bind to the hydrophobic trap are hydrophobic, which makes them not drug-like. They have off-target activities, like I mentioned, CYP inhibition, low solubility, high serum binding, and also limits the number of sites available for optimization. So the current status is, well, we're still working on it, but we haven't been able to solve these problems. And the current challenge is this CYP inhibition problem. And so when we decrease CYP inhibition, we find that we also decrease the metabolic stability of the compound, which I guess makes sense. And this is just a summary of what we've done so far and some of the future directions. So here's MBX 4191 divided into different zones by the medicinal chemists. And you can see that the zone 1 can't really change much here. This gem dimethyl group is essential. Zone 2, this cyano group is essential for activity. In zone 3, unfortunately, this sulfur group is also essential for activity, and we think it's a major site of modification by the cytochromes. The length of this chain carbon linker is also essential to position this phenyl group in place to make the pi-pi interaction with phenylalanine 178. However, we can modify the compound by adding different moieties to the para group of this aromatic moiety. And we've made several compounds, hundreds of compounds, where we've changed this group. And so far, we've found one compound that I can't talk about that does seem to prevent CYP binding. And then the other site we can modify is zone 4. Here we have a dimethylmorpholino group. This oxygen is actually important for making a hydrogen bond between water and glutamine 176. And so we've tried lots of different groups here, morpholine, piperazine, and adding different moieties to this oxygen group to make additional interactions. But so far we haven't found a combination between the modifications here and here that have the desired properties. So some future directions we're working on now is to explore alternative cores that maintain a hydrophobic character, but also simplify the syntheses of analogs. And also, so we've been able to find some alternative cores that are quite active, and also making macrocycles by linking zone 2 to zone 4 to zone 3 to restrict the compound conformationally, which we hope will improve stability and also improve metabolic stability, and also reduce CYP binding. So we managed to make several of these macrocycles, and some of them are active, and so we plan to continue because the macrocycles also offer the opportunity to improve the drug-like properties by modulating the linker that we use, by incorporating different moieties in the linker to improve solubility and stability. Okay, so I just want to acknowledge everyone who contributed to this work. A big team at Microbiotix, and a bunch of people over the years who are no longer with the company who've contributed to the project. Also, Martin Post's group for the crystallography work, Tilio Vargu, who did some molecular dynamics, Bill Weiss, and these guys, and also the NIAID for funding. Well, thank you.
Thank you very much. And that brings an end to the presentation part of this webinar. And I have to tell all three of our speakers we have quite a lot of questions. But for our audience, we will be doing 15 minutes of Q&A, and any of the questions that have not been answered, we will attempt to respond to you in writing. So all is not lost. So I'm going to start first with a question for Helen. The models you showed were developed for Pseudomonas. Do these predictions work for other gram-negative bacteria?
Depending on bacteria, right? So I think that prediction for outer membrane permeation would be working nicely with E. coli, because LPS layer and the structure of bilayer is very similar for Pseudomonas and E. coli. And also the major pump like MXB, which was optimized, right? So has high homology with ACRB and E. coli. So I expect that these predictions will be transferable to many enterobacteria, and we're checking this.
Thank you. So Ruben, got quite a few questions for you. One in particular was your resistance data that you shared. So you showed that in the presence of one of your inhibitors and antibiotic that you could get resistant mutants where they have a mutation ACRB. Do you think that would be a problem if they were ever deployed clinically?
Well, my view on antibiotic drug discovery is that it's very difficult to develop anything where there will be no resistance. The fact that we get resistance, I think, shows how clean the activity is. What is important is how easy it was to achieve that resistance. So in our case, it was around about 10 to the 8, 10 to the 9, which suggests there's only 1 or 2 residues that can be mutated that will allow you to get that resistance. But as you mentioned, it is possible to get this resistance. So it is always going to be there as a potential drawback, but I think one that is manageable, as it has got a similar frequency of resistance to other standard antibiotics.
Thank you. And one for Tim now. Clearly, you've spent a lot of time and effort making various analogues of your efflux inhibitors. Are these very hard to synthesize, and would they be difficult to scale up? A sort of generic question, I think this is.
Yeah. I'm not a chemist, but yeah, the pyridopyrimidine, pyranopyridine, our original scaffold does have some synthetic limitations. You have to make the core first, and then it limits the substitutions you can make. So yeah, and also the macrocycles are quite difficult to make, and could prove to be a problem for scaling up the compounds. But that's why we've started exploring other scaffolds to simplify some of the syntheses and to be able to make these at scale.
Thank you. So question for Helen, and I'm going to paraphrase the question from the person who's asked it. So they're talking about drugs that get into the host cell. So for instance, if they get in but are not easily effluxed, would they cause toxicity due to low excretion? And this person is particularly asking about the role of P-glycoprotein.
So it's a good question, right? Because there is certainly overlap between the specificities of eukaryotic pumps and prokaryotic pumps, correct? So I think that this problem is kinetic. So it depends on specificities and also how efficient pumps with this particular compound. And at the end, any compounds that we develop, it doesn't matter how it acts, will have to deal with host cell permeation and also host efflux. So, but again, so there will be balance of different features, balance of different specificities, and we just need to shift it into the right direction there.
Thank you. This is a question for Ruben. In terms of the inhibitors that you've synthesized so far, have you looked for potential secondary targets, such as are they exported by other R&D pumps or indeed other unrelated transporters?
So we've done a lot of work on the mechanism of action and understanding how specific it is. These point mutations that we identified were very useful in that, because those point mutations remove all activity of the molecule on the target, and we find that in the presence of those mutations there is absolutely no other activity. So it does suggest that in the E. coli system that we worked, where it's a little simplified, where there is one major pump, that is the main phenotype we see. Nonetheless, I do think that the other R&D pumps can be inhibited probably by the scaffold to a lesser, or maybe to the same or lesser extent, because the pocket itself is largely conserved. So there is a possibility that it would be active against other R&D pumps that are perhaps dormant or in other bacteria.
Thank you. I'm scrolling through the questions here. So Tim, what do you think is the greatest challenge in getting an efflux inhibitor and an antibiotic combination ready for first-in-man studies?
Yeah, I think that's a good question. Yeah, I think the big challenge is getting a optimized pump inhibitor that's drug-like, and then sort of developing it to. So I guess the big question that's come up is how do you administer the compound? Do you administer it as a fixed combination with an antibiotic, in which case you'd really have to match the PK/PD of the two compounds so that they're present in effective concentrations during treatment, or as a standalone inhibitor that you can administer separately with any antibiotic that the efflux pump inhibitor works with, and then you can modulate the you can match the dosing of the two drugs separately, so you're always above the effective dose in humans. So that's one major challenge. For us, the big challenge is turning these hydrophobic compounds into things that look like drugs. And so I guess those are the two things.
Thank you very much. So I'm really struggling to choose which one, because there's some really good questions here. So do forgive me while I just scroll through again. So Ruben, back to you. Do you think any of your inhibitors would need to have a universal tissue distribution? And I'm now combining this with another question. Could your inhibitors, with whatever drug they're combined with, be administered nasally? So direct into the lung, because you've got good lung distribution.
So I think the first question is a very interesting question. It's regarding the distribution of the compound in the body. And this is a very critical question for all of us developing efflux pump inhibitors. because infections are not always, depending on the bacterium, but are not always localized to single organs, and they can go from the lung into the blood or from the kidney into the blood, and therefore you probably need a good coverage, I would say, in either the lung and the blood or the kidney and the blood to treat the ailment or the infection. I think we had some very nice talks about that last week in the last Revive meeting on the target product profile. So this is something that we are conscious of. We think accumulation in the lung to some extent is very good to treat pulmonary infection, but we also need it in the blood to make sure that there's no sepsis that will come from any escaping bacteria. The second question was regarding to nasal administration. We have not tested that. Our molecules at the moment accumulate naturally in the lungs, but this is maybe not the case for all inhibitors of efflux pumps. So depending on whether it can be formulated and whether there is a market for such a formulation, that would be a possibility. One would, however, have to consider that in an ICU type of environment, I am not sure how easy it is to administer intranasally, and so perhaps direct administration into the lungs would be an option, but intranasally would still be challenging, I think.
Thank you. So I think this is for Tim, but I'm not sure. So Helen or Ruben, stick your hand up if you want to answer it. So if antibiotics theoretically interact with hydrophobic traps prior to being pumped out, what is special about the pump inhibitors that locks the pump conformation? Is it just that affinity is much higher for the trap or something else? So who would like to handle that?
I can handle.
Thank you.
So I think that it's a combination. So what we see, at least for Mpex series, right? So compounds that engage certain combination of residues within the pump have very different effect on whether they are going to proceed or stay longer on the pump and so on. So hydrophobic trap itself is important, but in addition to the trap, there are residues in the interface and in the deep binding pocket that also contribute to the binding of those inhibitors. So, and combination of all of them, including some of the specific residues, not necessarily hydrophobic, will define how long compound stays and whether or not it traps a specific conformation.
Thank you. So I was going to make that the last question, but I've got one more, which really just is one word answer from each of you is, if you had to choose an antibiotic to combine with an efflux inhibitor, what would be your choice? So I start with Helen, then Ruben, then Tim. So Helen.
Macrolides.
Any specific one?
Azithromycin.
Thank you. Ruben.
I think right now I would be going for a tetracycline.
Thank
or
you.
a, yeah.
Yep, and Tim.
Yeah, we were thinking about combining it with minocycline.
Thank you. So time is marching on, so we're going to have to move towards the conclusion of this webinar. So before I thank the speakers, I'd just like to advertise an inhibitor review, an efflux inhibitor review that will be coming out shortly by GARDP team. Also, there's a really interesting funding opportunity coming up from NIAID. The details will be in the link for everybody, and we've been asked to share that information with everyone who's in the webinar today. So that all remains now is for me to thank everybody, including the audience, excellent presentations, dynamic question and answer session, and we will be having another webinar soon within the next 4 to 6 weeks. So look forward to seeing you again. Do check our website, subscribe to our newsletter, and see our social media. So thank you very much, everybody.
Thank you.
Thank you.
Thank you.